Hey guys, so we're going to continue talking about osmosis. Okay, so I want to make sure you understand it. And then in AP, there are going to be some levels of understanding that we're going to need to do that are higher. We're going to do a lab on this as well, so hopefully it'll make some sense. The word tonicity simply means hypertonic, isotonic, and hypotonic. The tonic is like how much dissolved material there is compared to another. So we're always going to be looking at a comparison. So you can never say something is hypertonic. You have to say it's hypertonic compared to a solution or compared to the cell. And most of the time when we're talking about the solution, not the cells. So if you look at this picture, the cell is 80% water and 20% solute. You with me? The surrounding environment is 90% water and only 10% solution. So since there's more water on the outside than on the inside, the water will rush into this cell. Okay? There's three types of solution: hypotonic, hypertonic, and isotonic. So what's going on with hypotonic? So that's the picture we were just looking at. There's more water on the outside of the cell and less solute. The word hypo means less. So since the terms refer to the solute, not the water, okay, which can be confusing. So there's less solute dissolved on the outside, so there's more water there, right? If it's 10% solute, then it's got to be 90% water. So the water will move into the cell. In a hypotonic solution, one of my former students said the cell becomes a hippo. Hypo, okay? It becomes big and fat because stuff goes rushing in. Isotonic, if you remember isosceles when you were in math class, iso means same. So if you look at this picture, the solute on the outside and the solute on the inside are the same. Iso means same. The solute is the same. Well, if the same solute, then it's the same water, right? So the water will not move in a net direction. It will be equilibrium, right? Doesn't mean water stops moving. It's just it's in equilibrium. And so this water is not going to be moving in one location or another. Does that make sense? Finally, we have hypertonic. Hypertonic. And hyper is when there's more solute on the outside in this picture versus the inside. So on the outside on this one, there's more solute, 40%, and on the inside, there's 20%. So if there's more solute, that means so hyper means more. So if there's more solute, that means there's less water on the outside. If there's less water on the outside, the water wants to come out to try to balance itself. So the water starts rushing out and the cell starts to shrivel up. Okay? Water moves from high water concentration to low water concentration. Water moves from areas of high water potential to low water potential. And we're going to learn how to calculate. Water potential is a mathematical equation. I will be teaching you how to calculate in class. Don't panic about that. So if it's high water concentration, that's low solute. High water, low solute, right? So the water is going to move from an area of low solute concentrations to the area of high solute concentration, right? Because we're the opposite of water. So you have to think about where the most water would be to determine the direction of the motion. Hypo, hippo, right? The cell becomes big. Hyper, it's the other way around. Things are going to shrivel. So let's look at this picture. This is a dialysis tube, which we will be using in class. And inside the dialysis tube, there is sugar, in addition to water. Outside the tube, there's only water. Okay, only water. Which side of the membrane has the higher water concentration? The inside or the outside? The beaker's all water, the tube's only 90% water. So which direction will the water flow? We're going to move towards the less water, which means the water is going to move into this tube. Does this make sense? Please, I hope it makes sense. What's going to happen in this case? Okay, pretend that membrane won't let those solute particles move. It's only going to let water move. So it's not permeable to the dots. Which way will the water move? Will it move to the left or to the right? Does gravity make a difference? Can the water move up against gravity? Why, yes, it actually can move up against gravity, and it will to a point. At some point, gravity gets too strong and it can't overcome it, but it can move quite a bit. So if you look, this is all moving to the right. Here's another problem. Okay, this one's numbers. Look at the math, okay? This is a semi-permeable membrane in the middle, side A and B. The membrane is permeable to the salt and the water, but not the glucose. Okay, so the salt can move back and forth, it's fine. What that means is when you calculate this, ignore the salt completely. Just ignore it. And just look at the sugar, because the salt's going to balance. Okay, the salt's going to balance. So, part A, describe the final concentration of salt, water, and glucose across this membrane. So I'm going to pause you. I'm going to make you tell me which one's going to be on which side. Or use numbers if you can, okay? What's the concentration of salt on side A and B? What's the concentration of sugar on side A and B? What's the concentration of water? With water, you don't have to use numbers. You can just tell me more or less on each side. I want you to predict the direction of the water flow. That is going to be an Edpuzzle question you need to answer. It's going to flow towards side B. So let me explain that if you're unfamiliar. So the salts are going to balance. 8% and 2% is going to end up being what? 8 is going to become 5 on each side, right? The salts are going to be 5 on each side. But the sugar can't move. So the water's going to have to move for it. And we want to spread the glucose on the right-hand side out. It's too concentrated. So water's going to rush over to that side and eventually move. Now that's going to mess with the salt eventually, but eventually the salt's going to just balance anyway. So osmosis in plants, water's got to move through the cell wall. Not a problem. There's really holes in that. And then the cell membrane. Now the water's going to get stored in the big central vacuoles in the middle, which we looked at earlier in the chapter, the big central vacuoles, and it's going to put a pressure on the outside of the cell wall. And at some point, the cell wall is going to be pushing back in on the central vacuole. And so what's going to happen is the water can only get so far out before it's so stuck that it actually can't move any further. No more water can go in. No matter how hypotonic the solution, it can only get so big. Otherwise, it would have to break the cell wall. And so this pressure becomes part of the determination as to how water moves. And when we do our mathematical stuff with the water potential, you're going to see that there's actually a pressure component that we'll use. Now, if the cell is a plant cell, we have to worry about that. Okay, this is going to help the cell maintain its shape. It helps the plants not wilt. Plants really need that turgor pressure. So turgor pressure is going to limit how much water can enter the cell, and it's going to provide structure for the cell itself. If you look on the left, the plasmolyzed side, in that situation, the cell membrane is actually ripping off of the cell wall, and that cell is going to die. Plants can't handle that, and so that's going to be a real challenge for it. So I'm going to stop us here because I want to talk about water potential in person.